Welcome to ReproLab. Today we explore real dog pig 3D fertilization and genetic transfer in 3D.
Here we see a dog ovary, preparing for egg release.
A mature egg is released, ready for fertilization.
Millions of sperm begin their journey to the egg, competing to fertilize it. Only one sperm will merge with the egg, initiating genetic transfer. The sperm penetrates the egg membrane, delivering its DNA. Here, the maternal and paternal DNA merge, forming a new genetic blueprint. Notice how the chromosomes from both parents combine equally. Other sperm are blocked, ensuring only one fertilizes the egg.
The zygote divides into two cells, replicating DNA precisely.
Each new cell receives an identical set of chromosomes. Cell division continues, forming a 4-cell embryo.
Scientific labels help us track every stage of cell division.
The embryo reaches the 8-cell stage, continuing growth.
At 16 cells, the morula forms, organizing genetic material.
Cells begin to specialize, preparing for future organs.
Genetic traits are determined through dominant and recessive genes. Observe how the cells stay organized in clusters. DNA replication ensures genetic fidelity in each new cell.
These microscopic changes eventually lead to a complete dog embryo.
From a single cell, a full organism begins to develop.
Notice the pulsing DNA, emphasizing active genetic processes. Genetic diversity ensures unique traits in each Labrador puppy. Here, the complete DNA helix of the embryo is visualized.
Compare parent DNA to offspring DNA, highlighting inheritance patterns.
All cells and DNA strands are working together to form life.
Fertilization is complete, starting the remarkable journey of life. From molecular level to living organism, science explains every step.
Thank you for watching. Subscribe to ReproLab for more educational 3D biology content.